Good afternoon to everyone. So warm greetings to the Vedant Knowledge System Private Limited. So thank you for the opportunity as a session chair. So today I got some four papers related to biodiversity and related to microbiology and biotechnology, pharmaceutical sciences.
Namaskar and Pranam. Myself, Dr. Mehul P, and today I have selected a paper on a very nice, very interesting title, that is Civilizational Sustainability and Educational Reform: Ancient Indian Water and Soil Conservation in Journey Towards Viksit Bharat. So we want to understand that how ancient scientists in India, they were concerned and they were technologically advanced about the conservation of nature, biodiversity, forest, plants, animals. They were knowing various techniques of water conservation, soil conservation, and agriculturally they were very scientific. So we want to explore this idea. So in this presentation, we will check out those things. So, this is the title, friends. We want to understand civilizational sustainability, the ancient Indian blueprint for Viksit Bharat. So, this entire journey is very much relevant. That is what we want to say. And there was eco wisdom in ancient India. We want to dive very deep in ecological jurisprudence of that era in India, and which will lead us to carve a very nice road towards Viksit Bharat. So we had a lot of wealth of knowledge, including Sarve Bhavantu Sukhinaha, Sarve Santu Niramaya, Vasudhaiva Kutumbakam. We were very much concerned about well-being of all. That includes environment also. So Ekam Sat and Sanyam and Vasudev Kutumbakam, these were our cosmic philosophies. So in this paper, this is a very brief idea about the entire paper that I'm going to present today. So, ancient engineering, soil science, and how we were conserving water, we were handling data table, and what were the rules about the conservation of wildlife sanctuaries and Paryavaran and all that. So, the Western model and Indian model. So, Indian model is about Paryavaran, Pari and Avaran. So, humanity exists within the life support system, and we are much connected to the outer world. So, there is a strong relationship between within and outside us. So this is non-extractive jurisprudence model where, whatever I dig out, I should return back to the nature. That is the ancient Indian perpetual philosophy. So it's called reciprocity: give back what is taken. So in this paper, this will be the contents: ancient India and science, conservation of forest and wildlife, conservation of plants, ethnobotany. Animals were used as the vehicles of the deities, soil and water conservation, sustainable agriculture, and land management. So we have the legacy of Vedas, Upanishads, and Earth Shastra. Right? There are sacred groves and biodiversity hotspot written in all those ancient mythological books. So this is eco wisdom and our Paryavaran. And Indian sages has gave the mankind three tras: mantra, tantra, and yantra. So this is mantra, and then there was technology, Shilpa Tantra, and there were yantras for digging out water from the well, that is water mill. So our Vedas has every philosophy written in it about the nature conservation, and even Upanishads have. Indian scriptures have answer to all the modern problems, and they have the solutions to them. Right? Even if you read Surya Siddhant, then there is efficient calculation of the distance between Earth and Sun, very much measured. The speed of light is also measured, and distance between Earth and Moon is also mentioned, which is almost correct to the current scientific measurement. And even, Stephen Hawking has also told that Vedas might have a theory superior to Einstein's law, E is equal to mc square. Rig Veda, it also has the shlok yojanam sahasra dve dve shate, means it calculates what is the distance and what is the unit of the time and distance between Earth and Sun. And in Hanuman Chalisa also the same thing is mentioned: Yug Sahasra Yojana Par Bhanu. So there is a perfect calculation of the distance between Earth and Sun. These are our Vedic text: Yajurved, Rigved, and then Atharvaved and Samved. In Mahabharat, there is a concept of cloning, There is a concept of stem cell technology, test tube baby, and surrogate motherhood. Everything is written, and the greatest scientist was Maharishi Vedvyas. In ancient India, there is everything about nature conservation mentioned. In Atharva Ved, in Kautilya Shastra, in Manu Smriti, everywhere we bow upon the nature and natural resources. Right? There is a concept of Panch Mahabhut, Jal, Prithvi, Agni, Akash, Vayu. These are the Panch Mahabhut, and scientifically, this is very well correct. And it is written in Atharva Ved, Mata Bhumi Putro Aham. So the earth is my mother and I am her son. Even Lord Shiva is known as the Rudra, Satrudriya. He is known as the Lord of Himalayas also, and he is known as the Lord of the animals, that is Pashupati, and Lord of place, that is Vastospati. So there is a connection between the God and the nature. And then various animals are also mentioned in our ancient books, but here in this paper we are focusing more on water conservation, so we will directly go to that. But everything is written about forest conservation, including the pious and sacred trees like people, Ficus religiosa and Ficus caerica, that is fig. There were types of forest mentioned in our ancient books, that is Shrivan, Tapovan, and Mahavan. Even in Kautilya's administration also, there is everything written about the forest management, and there is Dravyavan, there is Astivan, there is Abhayaranya. Everything is very well depicted in them. There were two models of protection. One is Abhayaranya, and then sacred grove concept was also there in ancient India, right? And there were very much defined fine. When you cut down a tree, there is a different fine, and when you cut down a trunk and branch and the fruits, there were different amounts of the money that you had to pay. There is everything written about wildlife conservation also, and the king Ashok is known for issuing edicts. So in Ashok edicts also, it is written about the fine if you destroy the animals or destroy the even snakes and the various small insects also. Chatushpada and Dvipada, it is mentioned. So these are the sacred groves which are there in India, and we have connected the divine powers with the nature, so that the people will think twice before destroying any component of the nature. These are the current distribution of the sacred groves in our country, India, and they are much about biodiversity conservation. Vrikshayurveda is our ancient book. It is a medical text on tree planting and care, and there is everything written about plant life. It is ancient biotechnology and pathology, Bijopti Vidhi and Kunpajal, and then plant pathology, Tridosh, biofertilizer and seed priming. Everything is explained when we read Indian books, right? And then there is a profound science, as I mentioned, about restorative agriculture also. It's written in Vrikshayurveda. So species like Madhuka indica, they are also, they are revered. And Ficus religiosa, people tree, and banyan tree, right? In Maharashtra also, it is venerated. Soil and water conservation, it is very much mentioned in Rig Veda about water, and water exists in different forms. It is written Divya Sravanti and Khanitrimaha and Savyam Jaha and Samudra. In Yajurveda also, it is written water is nectar, medicine, and divine protector. Soil and water conservation, when we read the ancient text, we understand that. So these are the step wells which were prepared in ancient India: Jalar and Bavri and Khadi and Nadi and Tanka and Khadin. Right. So this is the entire IKS toolkit, but this leads our way towards Viksit Bharat definitely. So these are the types of the water conservation practices. It was hydraulic civilization. There was a perfect mastery over water. In Harappan legacy, in Mauryan engineering also we find. And then from hydro stress to hydro resilience. So this is the entire timeline which you can see. Indus Saraswati era and Vedic era and Mauryan era and Chola dynasty. Harvesting rainwater wherever and whenever it falls. It was the slogan. These are the engineering marvels in Shringarverapura and Dolavira about water conservation. These are the images of that. And we find every ancient reference mentioning that how precious water was for ancient Indian people. Johad and Tanka, they had different ways of storing water. And in Arthashastra, which is written by Kautilya, It is considered one of the earliest documents, and there is a lot of emphasis given on environmental ethics and wildlife protection. There is a codified conservation in Arthashastra. When we read the history, we find out that Naneghat in modern day Maharashtra is the oldest example of harvesting system and storage of the water. So this is the water harvesting down the ages in India. It is written Apaha Shanti, let the streams be at peace. It is written let pure water flow in our rivers. So when there is a modern water crisis and we think that next world war will be for water, at that time the Indian knowledge system comes to our help. The Dholaviran and Kautilyan synthesis, its anticipatory hydro resilience, Dholavira model and Arthashastra model. Now, we'll just explore little about the agriculture and sustainable agriculture and land management. And then Bhumi Pariksha was the practice. Germination test, flower test, and lamp test were performed by ancient Indian people. Crop rotation was the concept, organic fertilizer was found, and water conservation was very much efficiently done. We've seen. These are the lessons from ancient Indian conservation practices, holistic worldview, sustainable practice, community stewardship, spiritual motivation. So, this is the policy roadmap for a Viksit Bharat. You can see combined Khadin and Tanka with modern percolation tank, and there should be decentralized governance system. It is also found in Indian knowledge system books. And then restorative agriculture and ecological penalties. For any natural resource you damage, there was a policy, there was a penalty. So we have to redefine the development when we are having a vision for 2047. The world matrices were GDP growth, industrial expansion, and digital infrastructure. But over and above to that, we also have to consider ecosystem stability, community resilience, and intergenerational equity. These three aspects also must be considered when we have vision 2047. So we have to harmonize dharma with development, science with spirituality. Then definitely India will be Vishwa Guru, and it will be a Viksit Bharat. We have to think from exploitation to restoration. We have to be differently modern, and we have to inculcate and we have to engross the principles into our philosophy, into our engineering, into our governance, into our science. Because I would like to conclude that let us preserve nature and let us give respect to all the great sages and saints who have written a lot of philosophy in Indian knowledge system in ancient books and mythological scriptures. Thank you all. Thank you very much. This is the bibliography, the references that I have selected and read for preparing this paper. And these are the online resources in the form of bibliography. Thank you all. Thank you very much.
So among them, first I would like to discuss about civilizational sustainability and educational reform, which is presented by Dr. Mehul P. So Associate Professor, Department of Life Sciences, along with Dr. Deepika Patel, Department of Physics. These were from Indian Institute of Teacher Education. So ancient Indian water and soil conservation in the journey towards the Viksit Bharat. So the topic and idea is very awesome. And there we have the presentation is also good. Their abstract is completely analyzed with the study of the synthesis and strategic ideas of and importance of the Indian knowledge system. So I have an idea. According to this presentation, they want to make some clarity towards the education system towards the ancient to the modern Indian society. But they are missing a big points related what kind of the sustainability they want to make and what kind of the information they want to give exactly. So here they followed some kind of the review part related to the Indian history and philosophy and whatever the ethics and dharmas we are following. The system is good and the way of the presentation is good, but I have to understand what kind of the system and what a sustainability here they want to make. So according to the title, so they made it water and soil conservation in the journey towards the Viksit Bharat. So Indian culture and tradition is started long ago, and we are the torchbearer for the entire universe, and we believe in this way and how the system is going on. So apart from that, the presentation and article is awesome. They are written very well, and the holistic nature and biotic environment and whatever the secrets and roadmap, everything is they presented very awesome. So coming to the conclusion also, they written very good important points. So I wish all the best towards them, and coming research they can go for the better reviews and better way of the presentation. So thank you.
Good evening all. Today's my topic is regarding GCMS based phytochemical analysis and evaluation of the antioxidant and antibacterial activity of vetiver extract. Means we have taken this topic means in recent market Ayurveda and herbals are natural products are used in market for many things, and I am from the cosmetics background, and recently cosmetics in boom. So we have selected this topic and we have selected one extract like vetiver. It is giving smell also, and it has a very good antibacterial and antioxidant activity also. So from our sources, our Indian sources only, our plants only, we have taken this. So here the main objective is the GCMS analysis of the vetiver extracts. We have done extraction, plant extraction we have done, then DPPH activity we have done as an antioxidant activity, and antimicrobial activity of vetiver on the pathogens which are the presents in the as a smell odor causing bacteria. It's our natural normal flora only, but it is giving a smell when it interact with our body. So we have taken these organisms like Corynebacterium, and also we have taken this S. epidermis for this study. So zone of inhibition test also we have done for the Corynebacterium, and we found the 15 mm for the Corynebacterium with vetiver extract and 12 mm with for S. epidermis. So it has a very good antibacterial, antimicrobial activity against these bacteria. So we can claim that by using this extract in our routine, by using this extract we can make different type of the formulation also for body, which is good for anti-dandruff, anti-deodorant. So deodorant activity it will be given by this extract. So we are making some products also with this vetiver extract, but here we have the conclusion like vetiver extract is very good for the development of the deodorant part. We have done GCMS analysis, we got the good compounds also from that. We have done DPPH activity, we got the thymol and different type of compound this from this vetiver extract. We have done antimicrobial activity also with the vetiver extract. In the final conclusion, it has a very good antimicrobial activities, antioxidant activity. DPPH also we found very good in this. So it's very good for skin and for deodorant properties. Thank you.
So good afternoon. The second paper is on GSMS based phytochemical analysis and evaluation of the antioxidant and antibacterial activities of the vetiver extract. This is a plant extract. Basically this kind of the works are very awesome. These are the analytical works. But the authors, Mr. Hardik Bhatt, he concentrated more on the antimicrobial and antioxidant activity. But generally, this activity should be focused on the isolation of the compounds. So the way of his presentation is related to this one, but when he written everything related to the analytical components, how they can isolate and this one. So when the study is happening, so we need to go for basically with the TLC, thin layer chromatography, then we will go for HPLC, then only we can apply the GCMS technology. And the GCMS technology is not a final technology, even though they applied here and they got a very good result. And as it is based on that one, so they are given the too many compounds which is related to the chart based components and the availability of this one. But as per my knowledge, as per a reviewer of this paper, I would like to say that one, apart from making this particular activity, we need to write about the isolation of the polyphenolic compounds or the bioactive secondary metabolites such as the flavonoids and alkaloids, whatever the present in this particular extract. So after that you can test these for the antimicrobial activity and antioxidant activity. By chance they shown very good antioxidant and antibacterial activity, which resembles that one. This particular extract can be used as a medicinal plant. And the conclusion coming to this one, this plant or vetiver plant is root extract reveal the significant findings regarding the antibacterial and antioxidant properties, but the composition is also required. So what kind of the composition you are prepared, and what we are supposed to suggest the industry, like certain cosmetics industry or the pharmaceutical industry, the proportions we need to make according to the presence of the components or their chemical nature. So better to work on the same kind of the activity further to develop the industrial scale of the development activity. So thank you.
Namaskar, all the respected session chair and all the participants. I am Vaibhavkumar Pittaliya, postgraduate student at IITE Gandhinagar, Gujarat, India. Today I am presenting my research titled Comparative Analysis of Plant-Mediated Green Synthesized Silver Nanoparticles, Characterization, Stability and Antimicrobial Efficacy using the indigenous medicinal leaf extracts. Nanotechnology enables the manipulation of the materials at the nanoscales, resulting in the unique physiochemical and biological properties. Among the metallic nanoparticles, silver nanoparticles are widely recognized for their strong antimicrobial activity. However, the conventional chemical and physical synthesis method involve the toxic reagents and environmental hazard. Therefore, green synthesis using the plant extracts has emerged as a sustainable and eco-friendly alternative. So this approach integrates the indigenous medicinal plant knowledge with modern nanotechnology and support the vision of the sustainable innovation aligned with the NEP 2020 and the Viksit Bharat. Primary objective of this study was to comparatively synthesize silver nanoparticles using aqueous leaf extracts of Syzygium cumini, Azadirachta indica, Polythalia longifolia and Mangifera indica, and to evaluate their structural characteristics, stability and antimicrobial efficacy. So fresh leaves were collected from the IIT campus and proceed to prepare the aqueous extract. Silver nitrate solution was reduced using this plant extract under the controlled conditions. The formation of the nanoparticle was initially confirmed through a visible color change due to the surface plasma resonance. This was the first result. Second, the characterization was performed using the FTIR and TEM. The antimicrobial activity was evaluated using the agar-well diffusion method. So, by measuring that zone of inhibition against the selected bacterial pathogens. So, FTIR analysis revealed that the presence of the functional group such as hydroxyl, carbonyl, aromatic, and amide bonds, indicating that the phytochemicals including polyphenols, flavonoids, proteins, and metabolites played a significant role in reduction, stabilization, and capping of the nanoparticles. Another, that is the TEM analysis, demonstrate and predominantly spherical nanoparticles with variation in the dispersion and aggregation influenced by the phytochemical composition. The antimicrobial activity was tested against the Pseudomonas aeruginosa, Escherichia coli, Bacillus subtilis, and Pseudomonas species. All synthesized nanoparticles exhibit significant antimicrobial activity. Neem-derived nanoparticles from Azadirachta indica showed the highest inhibition against the Pseudomonas aeruginosa, while Polythalia longifolia demonstrated comparatively stronger activity against the Bacillus subtilis, and strong inhibition was also observed against the Escherichia coli and Pseudomonas species, confirming the broad spectrum antimicrobial potential. So in conclusion, this study demonstrates that the indigenous medicinal plant can effectively synthesize stable and biologically active silver nanoparticles through green synthesis method. The findings highlight that the importance of the integrating traditional plant knowledge with modern nanotechnology to promote sustainable nanobiotechnology and environmental responsible innovation. These plant-mediated silver nanoparticles show promising application in antimicrobial coating, biomedical materials, and etc. So these all are about my presentation. Thank you.
So let me explain about the third paper. So my reviews on the third paper, which is presented by Vaibhavkumar and Mr. Dr. Mehul P. So comparative analysis of the plant-mediated green synthesized silver nanoparticles, their characterization, stability, and antimicrobial efficacy using the indigenous medicinal plant leaf extracts, medicinal leaf extract. Here, the paper presentation is very awesome, and the activity and the research is also very good. And the growing science nanotechnology is very emerging now. And my intention to make the idea here. So, what kind of the silver nanoparticles and how they will be utilized, in which way they are utilized. So, the isolation and extraction and utilization is also very important. So, here the presentation is very awesome. The extraction process and methodology is presented very well. Characterization of the silver nanoparticles with which we got done by the quantitative analysis or the qualitative analysis. So, this analysis is not that much prominently showing here. So, which the photographs are not at all making any sense, and we need to write the table form or the graphical representation and quantification analysis. So, what kind of the exact binding is happen, and the TFM images also be explained a little more. So, by that the authors will get the good importance about the research activity, and this is a really awesome work. And I would like to congratulate the team to continue this kind of the works again, but with which we are doing somehow little clarification what we are doing and how to present it. So apart from that, this work is very good and the presentation is also very awesome. And I need this little configuration system. And other than that, this work is very good, and I wish all the best for the entire team of the who are working for this one.
Namaskar. Myself, Dr. Flora Shah. I'm working as Assistant Professor at Indian Institute of Teacher Education, IIT Gandhinagar, Gujarat. Today I'm going to present research paper on the review studies pertaining to molecular farming for phytopharmaceuticals, traditional medicinal plant-driven sustainable green bioreactors for affordable therapeutics, at the 5th International Virtual Multidisciplinary Conference on Reimagining Higher Education in 21st Century, an Inclusive Way Towards Viksit Bharat via NEP 2020 and IKS. Friends, as we know that there is a global demand for affordable biologics in the current scenario. The traditional system involving the mammalian culture aspects for production of the biopharmaceuticals requires very high cost and input of the high level of the infrastructure development. And hence, there is need of an ever for the development of the scalable and sustainable production platforms. In this specific scenario, the traditional medicinal plants involving sustainable platform systems can be working as the validated therapeutic sources for production of the phytopharmaceutical agents. And hence, the technique that is involving the biotechnological approach of the molecular farming can be considered as the best option for development of the biopharmaceuticals at the large scale. These systems are involving plant-based production of the recombinant therapeutics. They can be considered as the best alternative to the mammalian systems or the microbial systems. These are definitely working at the lower cost and they can be converted into the scalable platforms. They will be having the biosafety associated advantage also because of the reduced human pathogen contaminated risk. Traditional medicinal plants in this specific scenario can be considered as the best scientific assets. As we know that our indigenous traditional system of knowledge involving the traditional medicinal plant associated Ayurveda, Siddha, and Unani knowledge systems have validated specific type of the phytopharmaceutical components. They involve the curcumin with alloys, glycyrrhizine, and numerous other associated substances, which can be utilized very nicely for development of the target-informed metabolic engineering for production of the phytopharmaceuticals at the industrial level. This empirical validation is capable of supporting the translational research involving the biotechnological aspects with the molecular biology and the ethnopharmacology. For the molecular farming aspect for production of the phytopharmaceuticals, four major types of the plant extraction platforms are currently employed. They involve the stable nuclear transformation, chloroplast engineering, transient expression systems, and hairy root and suspension cultures. The mechanism of action of all these platforms differ greatly and they yield very good amount of the therapeutic production. The stable nuclear transformation involves the insertion of the desirable gene into the nuclear genome of the plant. They are capable of providing us the moderate quantity of the antibodies and enzymes as the therapeutic products. Chloroplast transformation system, on the other hand, involves the plant genome associated plasmid engineering. They are capable of providing high quality of the vaccine antigens as the therapeutic products. The transient expression system involves the viral vector associated therapeutic production of the vaccines and antibodies. Plant cell suspension cultures involve the culturing of the plant cells in the specialized type of the green bioreactors. They are working under the controlled environment and they can provide moderate to high amount of the recombinant proteins. Hairy root culture, on the other hand, involves the major mechanism of the Agrobacterium rhizogene induced root culture. They have the capacity of giving high amount of the secondary metabolites, especially the alkaloids and the terpenoids. For carrying out the specific culturing scenario, four different types of the sustainable green bioreactors are employed in the molecular farming technique. They are comprising of whole plant greenhouse systems, stir-tank plant cell reactors, airlift bioreactors, wave bioreactors, and hairy root bioreactor systems. With respect to the operationalism and the type of the phytopharmaceutical production, the whole plant system is considered as the most sustainable type of the technology because it requires the low energy input and involves the principle of photosynthetic biomass production and the vaccine and enzymes as the phytopharmaceutical products. Stir tank plant reactor systems involve agitated liquid culture. Here, we can have the advantage of achieving the controlled parameterization that can be developed at the industrial scale for production of large amount of the recombinant proteins. Airlift bioreactors, on the other hand, utilize air-driven circulation and can yield secondary metabolites in the medium to large quantity. Wave bioreactors are comprising of the involvement of the systems carrying out with respect to the rocking motion for the erection, and these studies are under the pilot scale. They are associated with the reduced contamination scenario and we can employ the transient expression system for phytopharmaceutical development. Hairy root bioreactors are utilizing the immobilized root culture systems and they can provide the high metabolite yield with respect to the phytopharmaceuticals involving alkaloids and phenolic type of the components. If we delve into the details of the competitive aspects of all the four different types of the plant expression systems utilized in the molecular farming, then we can say that transient systems are actually providing the fastest vaccine production to the pharma industry. Hairy root cultures can be utilized very stably for continuous synthesis of the secondary metabolite and chloroplast systems provides the high yield along with the biosafety. Stable transgenic plants are being employed for long-term protein synthesis. In terms of the pharmacological aspects, the plant-derived glucocerebrosidase are actually the approved type of the therapeutic agents which have passed the clinical trials. Vaccine antigens and monoclonal antibodies are under the clinical trials. We can take the example of the artemisinin and the paclitaxel type of the therapeutics which are accepted globally, which have been formulated using the molecular farming technique involving the plant-based production. Artemisinin is being produced using the engineered plant systems involving the metabolic pathway engineering and can be utilized to treat the malarial cases. They have commercially been approved at the clinical level. Paclitaxel is being produced using the suspension culture technology and it is the approved drug for the treatment of the cancer. Recombinant antibodies are being produced using the Nicotiana species and they are under the clinical trial and they can be utilized for immunotherapy. Vithinolloids are being produced using the root bioreactors in the hairy root culture system. They are under the preclinical trial and are utilized as the anti-inflammatory agents. Curcumin enhanced metabolites have been produced using the metabolic engineering. They are under the experimental stage and they can be utilized for providing the cancer treatment at the large scale. Plant derived glucocerebrosides have displayed comparable efficacy for the treatment of the Gaucher's disease. Virus-like particle vaccines have displayed strong immunogenicity in the phase 3 clinical trial for treatment of different types of the viral infections. Plant-derived monoclonal antibodies have shown favorable safety in the phase 2 clinical trial for treatment of variety of the infectious diseases. Vidania-derived extracts have shown symptom improvement in the clinical studies and they are being utilized for inflammatory disorder treatment. Curcuminoid associated formulations have shown improved bioavailability under the multiple clinical trials and they are considered as the best cancer agent therapy containing phytopharmaceuticals. If we go for looking into the beneficial aspects of using the molecular farming technique for phytopharmaceutical production, then we can find several economic and sustainability associated benefits. It involves the lower capital and operational cost. This technique is having complete absence of the human pathogen associated contamination and has you can say that it is most biosafety associated concern that is getting satisfied by using plant-based phytopharmaceuticals. It is involving utilization of the renewable energy in the form of the solar-powered biosynthetic machinery, and we can have the integration of this molecular farming technique into the agricultural sector so that we can have the economic upliftment of the country. In terms of the conceptual framework, the molecular farming technique involves the convergence of the ethnopharmacology and the plant biotechnology. It is associated with integration aspects of the knowledge of the molecular biology and the bioprocessing engineering also. It can be considered as the knowledge-driven target prioritization setting up technology involving the utilization of the sustainable green bioreactor for production of the phytopharmaceuticals. Still, there are certain technical and regulatory challenges associated with the regulatory harmonization requirement, plant-specific glycosylation differences, industrial scale-up associated constraints, and genetically modified organism associated perception and biosafety concerns. Still, the future of this molecular farming involving the phytopharmaceutical production can be considered as very bright in the course of time due to involvement of the CRISPR-based genome editing, glycogen engineering strategies, AI-enabled smart bioreactor involvement, multi-omics pathway reconstruction scenario, and traditional medicinal genomic integration. So in conclusion, we can say that molecular farming for phytopharmaceutical production can be considered as the best pathway towards the Viksit Bharat as it involves the conversions of the biotechnology and indigenous knowledge system of the traditional medicine. Not only this, it is involving the ethnopharmacology and biotechnology-led economic development of the country. It is capable of making the country susceptible and prone for production of the sustainable green biomanufacturing platforms for development of the therapeutic agents. It will lead definitely us towards the affordable phytopharmaceutical innovations leading to making the accessible healthcare solutions available at the global scale for all. With this, I would like to conclude my presentation. Thank you all.
So, this is my last paper for the review. So, it is presented by Flora Shah and Mehul P. And the way of this work is carried out is completely a review system and it's not a research. So, based on the data available, they presented this one, molecular farming for the phytopharmaceutical, traditional medicinal plants driven sustainability, green bioreactors for affordable therapies. Basically, according to the Ayurveda, Siddha or Unani, other medicinal things, and nowadays whatever the English medicine, allopathy, what we are using, we already have the different kind of the techniques. Most of the plant-based chemicals we are using for the extraction of the medicines, which is working on the plants, which is working for the human health and animal health also. And even till the date, most of the village people or the domestic nature, we are using the plant-related one. And the way of the written part and the presentation is also good. The thing is, if it is going with suggestion of the research, which kind of the methodology we can use for the analytical tools and the biotechnology tools, where this can be utilized for the medicines as well as pharmaceutical industry. So these are the development system, developmental biology and developmental pharmaceutical industry. These are very useful. But instead of this way of the review literature writing, we can do some kind of the experiment. So by that the results will be proven and we can grow for the plantation or we can suggest for any industry for furthermore and hybridization technology to get into the nursery or to get into the plant and agriculture basis and other advantages of this work. So I request the authors or suggesting the authors in the future, if there is any scope, kindly conduct the research oriented based articles. By that it will be useful for the society and useful for the industry. Thank you.